Bismillahirrahmanirrahim. Hello and welcome back, friends. Welcome to another video lecture by Kami Biology. So we already talked about the gram positive and we already talked about the peptidoglycan structure and basic overview of the bacterial cell wall. Now this is the last video lecture regarding with the bacterial cell wall, and this is the fourth part of the bacterial cell wall regarding with the cell. And in this video lecture, what we are going to talk about, we are going to talk about gram positive cell or gram negative cell wall. Gram negative cell wall. So gram negative cell wall, if we see gram negative cell wall, so if we see the gram negative cell wall in gram negative bacteria, it will be present between the two membrane, and this cell wall, or we can say this peptidoglycan layer, is thin. So means it is present between the two membrane. How? Let me draw here. So first of all, we have this layer of cell membrane. So this is the cell membrane, as you can see. So we have two membrane, we have two cell membrane. This one is called outer cell membrane, outer cell membrane, and this is the inner cell membrane, inner cell membrane. Between these two cell membrane, what we have the peptidoglycan layer. So these are what? These are the peptidoglycan layer. So in this case, the peptidoglycan layer will be thin. And in this case, if we see, the peptidoglycan are actually 2 to 4 layer. So there will be 2 to 4 layer of the peptidoglycan, and in gram-positive bacteria, as we discussed, there will be a 70 to 100 layer. It means that it is thick and it is thin. Okay, now this black one is name and this green one is neg, and both are connected with each other. So as you can see, this name will be connected, this will be connected with each other with a peptide linkage. So all these are connected with each other with a peptide linkage. Now as you can see the space here, this space, as you can see there, so this space are called what? Periplasmic space. Periplasmic space. Why we call it periplasmic space? Because it is not occupied by some proteins, as in gram positive. If you remember, in gram positive, the space is actually occupied by the thickness of the peptidoglycan, and the space is also occupied by the exoproteins. But in this case, the space is not occupied, so that's why we called it periplasmic space. Now one thing more, we have also a channel here. Let me erase this one portion. We have a channel here. How this channel will be look like? As I draw here, we have a channel just like that. We simply call that Porin's channel. Okay, so these are what? These are the Porin channels. Porin channel. If we see the Porin channels on top view, so how it will be look like? It will be look like in this form. These are actually connected with each other. Now this Porin channel can receive the molecules which have the size 600 to 700 Dalton. When the molecule have size 600 to 700 Dalton, then they receive this molecule. Like antibiotics, some of the antibiotic and some of the vitamins, means vitamin B12. So vitamin B12 and some of the antibiotics have a size like 600 to 700 Dalton, and they can easily receive these molecules. And once these molecules are received, then these molecules can perform their mechanism of action. So this is a little bit about the Porin channels. Now we have another important things. With this peptidoglycan layer. What we have a proteins like that, as I draw here. Now these proteins are called Burns proteins. Now after that, what we have some molecules arises here, as I draw with the black marker. These molecules are called lipopolysaccharide. What these are called? These are called lipopolysaccharide. It is actually, if we see, it is actually made up of lipid, protein, and sugar, means saccharide, or we can say sugar or glucose. If we see this lipopolysaccharide, so how this lipopolysaccharide will be look like? So it consists of three main parts: O side chain, core polysaccharide, and lipid A. So how these will be look like? So this one sides, as I draw here, these are what? These are the O side chain. O side chain. And then we have another chain with this polysaccharides, with this lipopolysaccharide. We simply call that what? We simply call that core polysaccharide. And it also consists of some like that chain. Now this chain is called what? This is called lipid A. Lipid A. So this is actually lipopolysaccharide. It consists of O side chain, core polysaccharide, and lipid A. Now this lipopolysaccharide is actually responsible to produce endotoxin in the gram-negative bacteria, which are specially responsible for the inflammation. And with the presence of this lipopolysaccharide, they can restrict the antibiotic and bisalts, which are harmful for the bacteria. This means this lipopolysaccharide are also responsible to activate the immune system as well. Okay, so that's all about the gram-negative cell wall, and that's all about the cell wall. So I hope you understand all these videos lecture regarded with the bacterial cell wall. Okay, so if still you have any kind of questions related with the bacterial cell wall, which contain 4 videos lecture, so write it into a comment. Inshallah, I will give you answer as soon as possible. So if you like this video, please hit the like button, like all the videos, and share all these 4 videos lecture with your friends, and subscribe the channel to get more interesting video lecture regarded with the cell. Thank you. Take care. Thank you so much.